So what we're going to do today, how things are kind of set up, is that we'll get through gustation and olfaction, and then we'll get into vision a little bit, eye and vision, and we won't get all the way through with that, and so we'll pick up with that next week and also finish up with ear, hearing, and equilibrium. So we'll go through and start in on eye and vision, then we'll take a quick break, and they'll tell you something about this taste activity, which is going to be kind of a low stakes type of thing, you know, where if you want to participate, you can. If you don't have to. All right. I did want to point out a few resources that I posted up on Blackboard for folks. So if you go to our the PowerPoints, in the PowerPoints, I have resources on for when we were talking about the brain, but I also posted them for the special senses. Right. So if you guys are tired of binge watching whatever it is that you're binge watching, you can binge watch, some of these resources. All right. In particular, this documentary is really interesting, Secrets of the Mind. It's about an hour long, and there's one section called Blind Sight, where people are not able to recognize the object, but they can tell you whether it's going up, down, side to side, which is really interesting. And so if you're curious about just honing in on that part of the video, I put down the timestamps for that. Okay? There's a companion site for that too. And then there's a lot of useful animations. Like I said, I think for the special senses, there's a lot of specialized structures within there that I think it's helpful to take a look at those animations. And so we might have a little bit of time to take a look at them in class, but in order to put things together and understand them a little bit more thoroughly, I really encourage you guys to take a look at these resources. All right. For the tasting activity, all right, there is extra credit opportunity, and you don't have to participate in order to get the extra credit, and so that's going to be this guy here. It's due next week. Okay? All right, and the directions are going to be found in here. All right, and so like I said, you don't have to participate in this in order to get the extra credit. All right? Okay. Okay, so before we move into special senses, let me ask, for what we covered last time, which was the autonomic nervous system. Were there any questions about that? Anything that was kind of fuzzy when you went through and took a look at some of that information? That was anything that was unclear? People were pretty comfortable with that? Okay. Well, if questions do arise, please seek me out so I can make sure to clarify that so you're not stuck, with looking at how that system functions. All right, so we're going to start off with looking at olfaction and taste or gustation. They're closely related to each other. The first one we're going to talk about is olfaction, which is the sense of smell. And some general flow of how we talk about the special senses is going to be, we're going to talk about some basic anatomy, just enough so we can talk about the physiology. And then we'll talk about the physiology, which is how these special senses, these organs function. All right, and so that's kind of general flow for each one of the special senses we'll talk about. So some general anatomy here. So we have our olfactory tract, which is going to end up in the olfactory bulb. So on the sheep brain dissection you all did, you may have seen these structures. Projecting out from the olfactory bulb are olfactory neurons. And so these neurons have their dendrites extending into the nasal cavity. So this is one of the few places in our body where nervous tissue is in direct contact with the external environment. These dendrites are kind of, sticking their processes, or the neurons, olfactory neurons, are sticking their processes in the form of dendrites out. On the ends of these dendrites, there's olfactory receptors. They bind olfactants or chemicals. And when a particular receptor binds to its cognate olfactant or chemical, it's going to trigger a signal. And so here, what ends up happening is that these dendrites send a signal, and then you get a synapse to carry, and then there's a synapse to carry, that message gets passed on to the next neuron that's going to send it to various areas of our brain. Now, is there emotional response to things we smell? Yeah, absolutely. So what part of the brain do you think some of these tracks are heading towards? What's that system called that's involved with emotion? The limbic system, absolutely. It's not the only place, but certainly anything that is arousing that kind of really gut emotion, that visceral emotion, it's going to be rerouted to the limbic system. All right? So you already know a little bit about how this system is being routed, how it functions. Now, it's not the only path that is being used to route this information. We'll talk about that in just a second. Now, one of the other things is that for smell, right? So how many folks are familiar with the phrase going nose blind? Right? So you get used to the smell. Right? So it turns out these receptors, when they get stimulated, they're stimulated for sustained amounts of time, right? They cease to generate a action potential after a while, right? So it means that that odorant or that olfactant hasn't gone out of that place. It's just that the sensory apparatus here, right, is no longer sending information for it, right? Okay, so one of the other things, back to talking about the anatomy, right? So it's going through this bone here. Anybody remember what that bone is? That's a funny name. Not the sphenoid. It sounds like sphenoid. Ethmoid. So I thought I heard it over here, right? So it goes through the ethmoid bone. So E before S. Right? So as you go anterior to posterior, you get ethmoid and then sphenoid. So if you remember taking a look at the ethmoid, there was little holes in it. Right? The little holes, the cribriform plate, that's where these olfactory neurons are going through. So if someone has a major impact that, say, breaks the ethmoid bone, right? What special sense is going to be majorly affected? Olfaction, absolutely. Right? So individuals that have fractures of the ethmoid bone can have major problems with olfaction. Now, what kind of hazards can come from not being able to smell? Oh yeah, smoke, right? So not being able to sense, dangers like smoke, right? So natural gas. Natural gas is actually odorless, so they add an odorant in there. What does that odorant smell like? Anybody know? Yeah, it's like sulfur, yeah, right? So if you don't know whether you've left your gas on, if you have natural gas, right, if you can't smell, right? So there's some major problems if you can't smell. Right? So one of the other things is for being able to do this adaptation where people goes nose blind, right? So let's say that there's a powerful scent that's in the corner, right? You walk to the center of the room, right? After a while, you can't smell that scent anymore. How can you figure out where that scent's coming from? Well, if you randomly walk around, eventually one direction you're going to start, that scent is a little bit stronger. And then you stop there, and then you're going to be nose blind again, right? And you walk around randomly again until you pick up that scent again. So how could that be helpful in the wild, you think? For like predators. Tracking, yeah, absolutely. Right? So this is something that is, quickly adapting these receptors. So going nose blind helps reset the system so you can hone in on where that scent's coming from, where it's stronger. Everybody following me on that? Does that make sense to folks? All right. So going nose blind isn't necessarily so that you don't know what's out there. It's actually so, it's thought to actually help you with being able to hone in to where that scent's coming from. All right. So olfactory neurons, like I said, are one of the few parts of the nervous system that actually contact the external environment. Okay? What ends up happening is that we breathe in air and it kind of gets turned around, so it terminates, right? And does so through, the conchae and so forth. So it churns up the olfactants so that those olfactants can bind to the receptors. All right, the odor molecules. Then you generate action potential. That's getting routed to different parts of the brain, including the limbic system. Okay? And so here you can see that, we have some of these olfactory neurons with their dendrites sticking out into the nasal cavity. Now, these olfactory neurons do get replaced, but they don't get the death rate of the olfactory neurons starts to exceed that of the replacement as we age. And so this is one of the reasons that as we age, olfaction, smelling, the ability to smell kind of decreases. All right. So which cranial nerve is involved? Well, it's going to be the cranial nerve number 1, the olfactory nerve. Right? So remember when we talked about cranial nerves, generally they go from 1 to 12 as we start anterior and then move towards the brain stem. Right? So here, if you're ever trying to guess which olfactory or which cranial nerve is involved with smell, with olfaction, right? Hopefully you're not going to guess 12 or 11, okay? So you can probably narrow down your choices to, 1, 2, or 3 in this case. All right, so processing smell, we talked about one of the routes that this smell gets routed to, or the place that it gets routed to, is the limbic system. All right. There's also an olfactory cortex here that's involved with processing, and then there's a neighboring association area for interpreting that. So how is that different from what we have going on in the limbic system? So when we talk about the cerebrum here, the cortex. So this is more conscious, like being able to identify where that smell is a Krispy Kreme donut, right? Or whether it is going to be sulfur that's related to gas. It's the conscious perception of what's going on. The limbic system is much more a visceral, response, right? So parts of the limbic structures include the hypothalamus and other ones, like the amygdala, for instance. All right. Any questions on smell there? Olfaction? Because we're going to move on to gustation, which is taste. And so here we look at taste buds. And so we're kind of starting off broad, and then we're zooming in, right, till we get to this taste pore here, where tastins or chemicals, right, are funneled into here, and they bind to a receptor cell. And the receptor cell then sends a signal to a gustatory neuron here. Okay? So it turns out, unlike the olfaction system, you don't have a neuron directly in contact with the external environment here. There's kind of, if you want to think about it this way, you can. There's a specialized taste cell that has receptors for certain types of taste molecules, and that's coupled to a nervous a neuron that's going to route that information to the brain. All right. Now we have different taste receptors that are going to trigger different types of tastes, and this mapping of which taste receptors on which taste cell, all right, is mapped in the brain. So like if this was a taste receptor for sour, right? Okay. When this whole cell gets activated and its neuron and its tract goes to the brain, it gets routed to a map in the brain and it says, oh, that's a sour signal. And so you get the sour taste sensation. Right? Everybody with me on that? Okay. So there's different receptors for different types of tastes out there. So it turns out there's basically 5 types of tastes that we have, or receptor types. We have sweet, sour, salty, bitter, and something called umami. And so not surprisingly, you can see that 3 out of these 5 are G-protein coupled receptors. The other 2 we'll talk about in just a second. The concentration of these different types of receptors is different in different areas of the tongue. So sweet receptors are more concentrated towards the tip of the tongue. The salty is a little bit towards the tip, and then it lateralizes, so it's a little bit more lateral. The sour is going to be kind of on the edges of the tongue. The bitter, which we'll talk about, is actually evolutionarily important, towards the back of the tongue. And then something called umami is covering roughly uniformly distributed over the tongue. All right. So I think people are pretty familiar with what sweet is. Okay. Sours are going to be acids. Salties, metal ions, so sodium, like in sodium chloride. Okay. Bitter, a lot of plant alkaloids are bitter. So kind of if you drink tea that's unsweetened, that's kind of has a bitter taste to it. That's the bitter that we're talking about. And then this umami is the protein component, right? So here, proteins that have that, kind of meaty taste. So this is one of the reasons that MSG is added to certain foods, is to stimulate this receptor. So remember, proteins are made up of amino acids. One of the amino acids that potently stimulates this receptor is glutamate. Right? The amino acid glutamate. So monosodium glutamate, MSG, potently stimulates this receptor, so you have that kind of meaty gustation. All right, so let's talk a little bit about how these receptors work. So let's take a look first at the salt and sour channels. They're actual channels. And so there's no really gating that goes on here. All they do is, all right, if, a salt, which is a sodium ion, remember we talked about metal ions, they're going to be positively charged. They're able to go through this channel. And if positive charges go into the cell, what does that do? What kind of polarization is going on? Are you hyperpolarizing or depolarizing? Depolarizing, right? Which means you're making it more likely to generate an action potential, right? So here, these work by having sodium ions or any metal positive ions go through these channels. And they are going to trigger a response that gets routed to parts of the brain that know that a sour receptor has been activated. Everybody with me on that? Okay. Now, if you remember, salt, not sour, salt receptor has been activated. The next one is sour. And sour has to do with the acidity. Now, if you remember, acidity is really the pH measurement, right? Is hydrogen ions or protons, right? H pluses. And so there's a channel that conducts H pluses through it. Now, just like with metal ions, if positive charges go into the cell, what is that doing to the cell? What kind of polarization event are we getting? It's the same as the metal ions. Depolarizing, which means it's more likely to generate an action potential, right? And so when these receptors, right, allow protons through, it's going to cause depolarization, and therefore you're going to get this signal sent to the brain. The brain knows that, if this signal is coming from this track, it means that this sour receptor was activated. Okay? The brain knows that if a signal is coming from, say, a different track, a different receptor was activated. For instance, a salt receptor, right? One with the metal ions. Everybody with me on this? The tracks are different for different receptors, and the brain has a map of that. Okay? All right, so the other ones are all G proteins. So here, sweet, bitter, umami, they're all G proteins. And since you guys are experts in G proteins now, I don't have to go into huge detail about this. But here, before we have that ligand or that tastant binding, we have an inactive G protein, which means which type of nucleotides bound to it, GDP or GTP? GDP. And then when we have that tastant bound to this particular receptor, now we have an active G protein, which means that which type of nucleotides bound to that now? GTP, absolutely. And so we get second messengers that are going to be sending signals or generating an action potential that is going to be propagated to different parts of the brain for interpretation, right? So if a umami receptor gets activated, that is going to be known by the brain. Okay? Everybody with me on that? Okay? So those are the 5 major taste receptors and their distribution. Yeah, go ahead. Mm-hmm. Yeah, no, that's a great analogy. The leakage channels, because they stay open. Absolutely. Yeah, fantastic analogy. All right, so where does this get interpreted? Right? Well, it turns out the gustatory cortex, where you have conscious, realization of what the taste is in your mouth, is in that 5th lobe that's called the insula. So here we have the parietal lobe up here, and then here's the temporal lobe. And remember, separating those two was that lateral sulcus, right? So this is a coronal or a frontal section. If you pull back that lateral sulcus and peek inside of here, you see this area, which is the insula, right, where we have the gustatory cortex. Okay, so you can see it pretty nicely here. Now, what routes or what types of cranial nerves are involved with routing this information to the brain? Well, there's 3 cranial nerves involved, right? Facial and glossopharyngeal are 2 major ones. The vagus does some in the back of the throat, okay? But all 3 of these are routing information to the gustatory cortex. Now, do you have any visceral or emotional feelings when you eat something? Yeah, probably. So what's another place that some of these, pathways route to? The limbic, right? Okay. So there's going to be other, so I told you about, say, the bitter receptor, right? Okay. So bitter receptors detect in minute amounts. So what were bitter receptors really good at detecting? What was the example that I gave you guys? Plant alkaloids, right? Plant alkaloids. Turns out a lot of alkaloids and, poisons are pretty bitter. So why would it be beneficial to detect in minute amounts compared to other receptors or other tastes? So you don't die. Yeah, absolutely. So you want to be able to detect, minute amounts so you don't take enough to cause your body harm. The other thing is that this is also closely related or tethered to the gag reflex. Where in the distribution were those bitter receptors? Where on the tongue? In the back, right? So think about the gag reflex, right? So here, the bitter receptors and also a gag reflex, are closely tied to each other. So how is that beneficial with what I just told you guys about? Right. Yeah, you get it out of your system. You get it out of your system, absolutely. Now, I always wondered how they wrote people into these studies to figure out whether poisons tasted bitter, right? Poor graduate students who probably don't have much money, or students in general, they say, hey, we're going to give you some poisons. You tell us, what they taste like, right? And it probably didn't give them enough, to cause them harm. But I always find that interesting. I would like to see the flyer that was made to rope people into that study. All right? So here, this is really beneficial because a lot of those plant alkaloids, right, poisons are bitter. Okay. And so you can see where these cranial nerves are innervating different parts of the tongue, in some cases different parts of the throat structure as well. All right, so basic process for how we taste is that we get food and we're going to digest it a little bit in our mouth to release some of those molecules so that they can either bind to the G protein coupled receptor or passage through those channels that are constantly open, right? And so that's what's happening in these first few stages here. And then they're going to attach to those receptors. And because they're chemicals, these are called chemoreceptors. So what types of receptors were olfactory receptors? Chemoreceptors as well. They're attaching to chemicals, they're being activated by chemicals. So far we've really talked about, if we want to classify these, we're looking at chemoreceptors. And you get that local depolarization that can lead to an action potential, right? Now, remember, what's the difference between olfactory, the cells that have the olfactory receptors versus the cells that have these gustatory receptors? Yeah, exactly. So you kind of have a receptor cell here with the gustatory sensations, as where with the olfactory receptors were directly attached to dendrites of the olfactory neuron. Right? So the olfactory neurons having a direct contact to the external environment is one of the few places we actually have nervous tissue in contact with the external environment. All right. So it turns out 80% of taste is smell. So you can do this experiment on your own. So you can actually hold your nose and then put something in your mouth. And then if you release your nose, you are able to taste it a lot better. Right? So you can do that, with, say, random, if you trust somebody, like random Skittles and try to guess what flavor it is. You can't do that guessing very well with the nose unplugged. As soon as you unplug your nose, it's a lot easier to do that because 80% of taste is smell. Right. Now there's other receptors that we have in our mouth and our tongue. So we have touch, pressure, temperature, pain, also influence the taste experience. So textures. So there's some cultures where the texture of food being what we consider slimy is preferred. It's something that is sought after. Right? It's not here in the Western culture, especially here in the U.S., but in some cultures it is. Right? And so here, textures influence, the taste experience. Additionally, some drugs influence the actual gustatory processing that goes on and the actual taste receptors themselves. So there's some drugs that interfere with conduction of taste receptors. A lot of some chemotherapeutics for treatment of cancer can interfere with the taste sensation. Right? So there are pharmacological compounds out there that can muck up this process. All right. So just before we get into eye and vision, I'll tell you a little bit of one of the researchers who was doing research on taste receptors. And he was looking at G protein coupled receptors. And remember I told you that the receptors are the track that the receptors send their information towards the brain is mapped in the brain, right? So what he ended up doing is taking one G protein coupled receptor and another G protein coupled receptor and swapped them. right? So he took, say, the bitter receptor and the sweet receptor, swapped them in mice, right? So now you feed mice something really, really bitter. What pathway is getting activated? So we switched the bitter and the sweet pathway. So now they're thinking, wow, this is really sweet. I want more of it, right? And so here, really bitter things, they were just going bonkers with, hey, I can't get enough of this, right? And opposite for sweet things. Yeah. This is kind of an interesting study. I thought I'd share with you guys, right? Okay. All right. Any questions on gustation there? Does that make sense to folks? How it's similar and different from olfaction? Feeling pretty comfortable? Oh, all right. Okay, because we're going to get into a little bit of vision here. And the eye, we'll first talk about some basic anatomical structures. You're going to see this again in the lab, right? So I don't think it's ever a bad thing for you guys to hear or see anything twice. Okay? All right, so we have the eyebrows and we have eyelids, which are known as palpebrae. And so we also have some muscles that are controlling some of the external structures of the eye. So external structures from the eye we call accessory structures. And so here some of those are levator, palpebrae, the superioris, orbicularis oculi. So all the different things we can do, not with moving the eye, but moving these accessory structures around the eye. You know, some of the muscles I've listed here. Now, the muscles that are actually moving the eye are called extraocular muscles. And remember that mnemonic device for knowing which of the three cranial nerves are involved is OAT, OAT. So you have the oculomotor, the abducens cranial nerve, and the trochlear cranial nerve, right? So 3, 6, and 4. Turns out that these extraocular muscles, so if we have the eyeball that looks like this, you have one extraocular muscle that is attaching to the top of the eye, one to the bottom, and then one to the medial and one to the lateral. So if this is my right eye, okay? Right? And then we have another two that are going to be diagonal. Okay? Actually, they come back like that. Diagonal on the top and diagonal on the bottom. So those are the 6 different eye muscles. The 4 that are on each side of the eye are done by this oculomotor. The abducens is the diagonal one that's on the bottom. The trochlear is the kind of the diagonal one that's up on the top. So we got 3 here, another one here, another one here. So, 4 here, another one here, another one here. Total of 6. Okay? So why are those 2 diagonal? Well, it turns out those, the superior rectus, which is on the top of the eye, and the inferior rectus, turns out when you look up, it actually pulls the eyeball a little bit lateral. And to offset that, we have these diagonal ones to counter that pull, right? So these diagonal ones counter that pull, right? And so I've got a few resources for you guys to take a look at up on Blackboard if you're really interested in taking a look at that. Okay? All right. So some of the other structures, conjunctiva, you guys may have heard about, is a clear membrane that covers the surface of the eye. The very anterior part of this is actually something called the cornea. All right? So conjunctiva is clear, and you can see it's this kind of sheet, and it folds back along the interior surface of the palpebrae. When this becomes inflamed, we call that conjunctivitis. Okay, conjunctivitis. So a lot of times it comes inflamed because it gets infected. So pink eye, conjunctivitis. That's what we're talking about. This membrane's getting inflamed. All right. So looking at the lacrimal apparatus, tear formation. So superior and lateral is where we have the lacrimal gland. And so here we get the secretions that come out from superior and lateral. And they wash down in this direction across the surface of the eye. Now, where do they exit? They exit here in these lacrimal puncta, and there's little ducts that carry it to this sac or in this canal that empty into the nasal cavity. Right? So if you take a close look at your eyes, when you get a chance, you'll see two small red dots there, and those are the puncta, where the secretions from the lacrimal gland get evacuated, right? So what happens if we produce more lacrimal solution, tears, right? Tear solution, then we can remove. It overflows our eyeballs, and that's tearing, right? So what are some ways that can happen? So one way is to generate a lot more tears, right? Tear solution. What's another way that can happen? So block this area, right? So if these puncta get blocked, right, that can also cause problems. Now, when you have evacuation of tears into the nasal cavity, what does that look like? So think about kids when they're crying. Snotting up, right? So sniffles, right? So you see uncontrollable crying, with children. A lot of times you see the sniffles accompanying it because there's huge dropping of liquid from the tear solution into their nasal cavity. That's what's going on. Right? Does that make sense? Kind of the flow of things. What's the purpose of this? Why have it at all? What's that? Clear debris, yeah. Keep it clean. Yeah, so there's a lot of stuff in the tear solution to help keep it clean, right? And if it's constantly washing this way, it's constantly removing things, right? All right, so here's a sagittal section of the eye, and some of these things we have taken a look at already. You can't really see the conjunctiva too much, but you can see the cornea here, okay? You can also see a chamber where it has liquid in it, and then we have the lens, and then we have another chamber with another liquid filled chamber, I should say, right? So here, this large liquid filled chamber is called the vitreous chamber because it has a vitreous humor. So fluids are generally called humor. All right, so three major layers of the eye. The outermost is called the sclera. You'll see this when you get into lab because you guys are going to be doing a steer eye dissection. This is a tough layer. Why is it tough? Well, where are the extraocular muscles attaching? Onto the sclera, right? So if this wasn't tough, then you wouldn't have ability to pull as strongly as you need to move your eyes. And so here we have the sclera. That kind of ends with, the cornea, which is clear here on the front end. Underneath the sclera is something called the choroid, this reddish layer here, right? That choroid has a lot of blood vessels. It provides a lot of nutrients for the next layer that we're going to talk about, all right? So here, the choroid, if you follow it anterior here, it turns into what we call some of the ciliary muscles, and then also the iris. So the iris is a colored part of our eye. The hole in the middle of the iris is the pupil. Okay? So a pupil is just an opening of the iris. So we get our different iris colors. They are determined by how much melanin individuals have. So lighter color eyes have less melanin. Darker color eyes have more melanin. All right? Okay. So the internalmost layer is one that's called the retina. The retina has the sensory apparatus. it gets all of its nutrition from the underlying choroid because that has all the blood vessels. All right? The choroid is also heavily pigmented, and we'll find out why that is in just a second. But the retina is the anterior of the eye, and it has the photoreceptors, and the neurons, the axons of those neurons, eventually exit into the optic nerve. All right? Now, there's a part of the retina where you have a little depression. It's called the fovea. And this is where we get our highest resolution. It has a high concentration of what we call cone receptors. Okay? And so that's where we get our highest resolution. All right, so that's just some of the basic structures of the eye. If you kind of look at the back of the eye, right? So here you can see the optic disc. That's where the cranial, the optic nerve is exiting from the eye. So if I go back a slide here, right? So that's this. Okay? So it turns out that these are just axons exiting. They don't have any photoreceptors in here, which means if light hits this part of the eye, there's no photoreceptors in order to detect it, which means that this is really a blind spot in our eyeball because all it is are axons exiting the eye. So what does our brain do? It does some sophisticated modeling to try to fill in that blind spot so you don't constantly see holes. Okay? So here, this optic disc is also known as the blind spot for that reason. There's not really photoreceptors to detect the light that hits that area. Okay? Now here you can see the macula lutea. So if you think of this as a target, macula lutea, that's giving us high resolution. The highest resolution is the bullseye within that target, the fovea. Okay? Sometimes it's called the fovea centralis, right? So the fovea is kind of the bullseye, and the macula lutea is the target, okay? And that gives us the highest resolution. So a lot of times when we're trying to get high resolution, you don't know this, you're probably not consciously aware of it, but you're scanning to try to get that part of what you're looking at hitting the fovea, and then your brain stitching together all of those series of high-resolution images.
Is that why we squint, like we're not wearing my glasses and trying to see some pictures?
You're also maybe distorting the eye a little bit, to try to do the correction, is another reason I think, that squinting happens, But generally squinting also happens when you're trying to keep stuff out of the eye, So, yeah. Yeah, and this is a little bit more unconscious. You're not really aware of it. Doing the scanning, and then your brain stitches together these high-resolution images to give you, this field of view. All right. So here we talked about the retina. That's the neural part. Has the photoreceptors. Rods and cones are the two types of receptors we have, and then we have the optic disc and the fovea within there. Right. Now, if you take a look at this, you see the blood vessels here. Right? So what layer was that? The choroid. Absolutely. So how many folks have seen, like in those movies where they're trying to get into a really well-protected vault or something's protected and they do retinal scans, right? So what is that doing? Well, it's they have on file the pattern of the blood vessels that are in your retina. Each one is different. That pattern's different for each person. So it's called like a fingerprint. So what is it doing? It's matching whatever the pattern is that they got with what they have on file. Right? So they're saying, okay, does this retinal, blood vessel pattern match who this person is? So that's how that retinal scanning works. So that they're looking at this pattern distribution of retinal blood vessels. Right? Okay. So there's one thing I wanted to say about this, and then we'll take a break. The cornea. The cornea is avascular. So what does that mean? No blood flow. Does that mean that the immune system is going to have access to that? No. So we can say it's, immune privilege. So one of the reasons that corneal transplants can happen without finding, say a specific match is because the host, the recipient, okay, is not going to be able to reject a cornea because their immune system isn't going to be able to detect whether it's self or non-self, right? So corneal transplants can happen without too much of an issue because there's no real intervention by the immune system because the immune system is somewhat blind, right, to that. Does that make sense to folks? Okay, so I think what we'll do before we get into physiology of vision, let's take a 5-minute break, and then we'll do the taste activity, and then we'll pick up with physiology of vision next week.